All right, so polarity. So polarity, I would say, is really one of the universal features of biological systems. So polarity is already, hugely apparent at the molecular level, right? Usually, even like simple chemicals will have two distinct ends. Think about DNA nucleotides, for example, that polymerize because they have two reactive groups at either end. So they polymerize in a certain direction, generating polarity that way. Polarity is, of course, also an essential aspect of the cytoskeleton in the cell. So, for example, microtubules that drive a lot of the biology that we talked about this week, they are made up of these dimers of two proteins, and already you can see that the dimer itself already has a polarity axis, and then these large macromolecular fibers are assembled by the head-to-tail alignment of these dimers. So the entire fiber, therefore, gets a polarity, which can then, for example, be translated into directional force or directional flow by motor proteins walking along the fiber, along its axis of polarity. Second, polarity is, of course, also apparent at the level of cells. So here I'm showing at the top, this is an epithelial cell layer, so basically something you could think of as lining, for example, our lungs or our intestines. And these cells have functionalized differently the membrane that faces the outside versus the membrane that faces the inside. And that, again, is hugely important for biology. Also, neurons are an example of a highly polarized cell type, right? So they have this around their cell body. They have the dendrites by which they integrate information, but then they grow out this long axon that transmits the information over long distances. But then polarity is also obvious at the tissue level. So, for example, think about the scales in a fish that are all aligned along the long axis of the animal, or the famous wing hairs on the Drosophila wing, that if you were to look closely, all the wing hairs on the wing will be pointing in consistent directions all along the tissue level. And then finally, polarity is also obvious at the organismal level, because we generally tend to have a head in front and a tail in the rear, depending on what vertebrate class you belong to. But needless to say that also the body axis of all multicellular animals is polarized. So this then, I think, makes it fair to state that polarity is in a way a design principle of biological systems, and it is apparent at all scales of biological information. Now, what creates polarity, or what are the machines that create polarity? So this is actually best understood at the molecular level. And so, for example, in case of the apicobasal polarization of epithelial cells that I mentioned on the previous slide, what is happening there is that actually at different levels of this lateral junction between cells, you will have different protein complexes localized there. And so the specific details here don't really matter, but what is important to note is that these complexes both interact with each other. So basically the ones that are sitting up here, they will tend to repress the ones that are down here and vice versa. And secondly, these complexes also couple then to adhesion molecules that connect the neighboring cells. And this is very important because in this case, these complexes or the junctions here, they actually make tight diffusion barriers between cells, which is, for example, required for the epithelium to separate outside from inside. So in this case, polarity is essentially set up along the apicobasal axis of the cell. However, polarity can also be set up along the mediolateral axis of cells. And in this case, it is the so-called core PCP pathway that is one of the key players in bringing this about. And again, just imagine now you are looking down on a fly wing from above, and what you would actually see, or how planar cell polarization would manifest at the cellular level, is essentially that you have two different complexes, protein complexes. Sorry, the colors don't really match here. But you can, so basically localized on opposite ends of the cells. And the way this comes about is that within each cell, there's essentially a set of proteins that, again, mutually inhibit each other. So this means that they will naturally tend to segregate in the membrane, right? So you can think of it either on one side, there will be the blue complex, and on the other side, there will be the green complex. And now, again, because these proteins are membrane proteins that have extracellular domains, and these extracellular domains couple again to the opposite complex on the opposite side. So for this reason, you therefore tend to end up with these staggered and nicely organized sequences of polarity protein complexes within individual cells and also at the tissue level. And now, interestingly, this machinery here, the proteins that are involved in that mechanism, they actually are also partly shared with this wind signal transduction mechanism that I talked about yesterday that is so important for specifying head versus tail in planarians, because the receptors for the wind signal, some of them, they are actually also part of these differentially segregating complexes in planar cell polarity. And second, this intracellular transduction protein, disheveled. This is also a key player in the planar cell polarity pathway. And so again, this is probably actually, there's some deep meaning behind this, so that this signaling pathway can do both cause state switches, as in the scenario that we talked about yesterday, as well as specifying directionality within the tissue via these neighbor-neighbor coupled systems. But there are still lots of open questions here. And then, so this classical core or core PCP mechanism is not the only one that can generate planar cell polarization in the tissue. But a second pathway that is actually quite well studied is the so-called fat-Daxis pathway that is also operational in the flywing and mediates mainly the radial polarization of the disc from the outside in. And this also, again, operates on very, very similar principles to the core PCP machinery. So again, having essentially mutually inhibitory membrane components that also that segregate within a single cell, but also interact with the neighboring cell in trans. And so altogether, I think there are two important points here. The first one is there are many polarization machines in cells or multiple mechanisms that can polarize cells. But in general, they all share this sort of cell autonomous polarization component, right? That individual cells can set up a vector of polarity. And secondly, they share this neighbor-neighbor coupling effect, so that basically the polarity vectors of individual cells get coupled between their neighbors and therefore set up polarity fields in the tissue. And this is actually, on a conceptual level, this has been very much likened to ferromagnetism, for example. And so for this reason, also there have been many models of PCP basically of PCP systems in biology. And this also continues, or modeling and theory continues to be also a very important driver in this field. All right, so now, very good. Individual cells can polarize. Their polarization can be transmitted to neighbors. So what is this good for? Well, all the cells are would be polarized. Touch the other cell, polarized. So that would obviously mean that this mechanism cannot work. However, what could happen under these conditions is that maybe there's a memory component to it, right? That the cell at an earlier stage where it is in a field where the neighbors also express PCP components, that at this stage it becomes polarized by the neighbors and then maintains this polarity by some other mechanism. So that could be a possibility But I'm not familiar with the organ of Corte, but that's an interesting case, so I will look into this. All right. That's, you know, essentially that's the essence of the question. Well, or that's one of the questions in the field is basically what initiates the polarization. So is this what you are getting at, or exactly? So that, because you could imagine that initially everything is sort of in an unpolarized state, and then something initiates the polarization to spread through the tissue. This is, you know, in many, so what happens very often in development is that the polarity is specified very early, and then it is maintained during growth. But there are also other systems where there's a clear symmetry breaking event that then initiates the polarization of the cell field. Well, so I think actually, you know, in few cases, or I'm actually not aware of a single case where this has been, you know, where really molecularly it has been taken down all the way to the molecular level to explain what is happening. But so a standard paradigm, for example, would be in a Drosophila wing that is already planarly polarized to induce a clone of cells that, for example, are lacking frizzled. Right, so they are basically, they cannot couple to their neighbors via this thing, and then what will happen is that they will non-autonomously repolarize the cells around them on one side. So in this sense, you could say that by, you know, by changing by changing a patch of cells and making them basically blind to these interactions with their neighbors, they also influence their neighbors again and also repolarize basically the area around them. So, and in general, you could also think of an origin of polarity in the system, or well, basically the cells themselves cannot be unpolarized simply because these membrane complexes will never mix with each other. So they will always form separate patches in the membrane. And so therefore, in a way, so, you know, to have a tissue that is naive in the sense that cells are unpolarized possibly cannot exist, or to my knowledge also has not been demonstrated. But yes, lots of, you know, questions that are definitely legit, and just because we don't see it in biological systems, that's why biology doesn't really or cannot really study it. But of course, from a theoretical point of view, or also from a conceptual point of view, this is really also part of the essence of the problem. All right, now, yep. Well, so, you know, this is again, in some sense, the same answer. So in biological systems, generally polarity happens very, very early on, or polarity is specified very, very early on. So in these cases, you know, it is really difficult to see sort of unpolarized states of biological tissues. That's why it's a bit difficult to, you know, to answer this question. Okay, so what is this polarity good for? At the tissue level, for example, there's the alignment of hairs in the Drosophila wing that we already mentioned, but you can actually demonstrate exactly the same thing in mice. So basically, if you take out components of this planar cell polarity pathway in mice, then also the fur of the mouse will start to develop these characteristic swirls. So exactly the same phenotype that you also see in Drosophila when you take out components of the system, where then essentially the long-range vectors start to break down and you start to see sort of localized disturbances, so basically swirls in the polarity field. What these planar cell polarity mechanisms also do is that they orient cilia. And so, as you know, cilia are these basically long extensions on the surface of cells that can beat and therefore are very important for actually generating flows in biological systems. Now, because many or common type of these cilia, they will beat forward and reverse, so to speak. And in doing so, therefore, if you want to generate a flow field in tissue either with many cells or with cells that have many cilia, it is clear that because of the directionality of the beat of the cilium, all the cilia have to beat into the same direction in order to achieve consistent flow. And this again is achieved via the same planar cell polarity mechanisms. So where essentially now the protein complexes on the side of the cell, they couple to the base of the cilia in order to orient them in a consistent manner. And finally, and this is also what you already heard about this week, these planar cell polarity pathways are also very important for coordinating the dynamic flows of cells in such a way as to, for example, mediate intercalation during development. And if you, this here is a zebrafish in which the PCP has been disrupted, and the tail of this animal is short, simply because the cells can't intercalate, and therefore axis elongation is basically stunted in this thing. So, long story short, the planar cell polarization of cells in the plane of the tissue is actually very important for the long-range coordination of cell behaviors. Now, so in the case of the fly wing, no. These flies can fly just fine and, no problem. So it was clear, why was it selected for? Why are the fly wing hairs oriented? Is this directly adaptive or not? Probably not. In the case of the mouse, also the fur pattern probably doesn't really have a specific adaptive value. However, this here convergent extension is also hugely important in a mouse. So you won't get an embryo without PCP. And also the cilia alignment is very important in the mouse. So basically, if PCP is defective, these mice develop, or the common phenotype is, for example, hydrocephaly, so the accumulation of fluids in the brain, simply because the flow doesn't work. So long story short, a mouse with a PCP mutation everywhere would be dead and would never be a mouse. And whether then the specific manifestation of PCP in the epidermis, whether this is a sort of a system consequence of some other important function of PCP, or whether it's directly adaptive, that's one of the questions that would be very, very difficult to answer in nature, actually. All right, so now, this is what PCP does, challenges of PCP. And we already talked about quite a few of them, actually, now. The first one is the origins of polarity vectors, right? And because we are dealing with this intrinsic polarization and neighbor-neighbor coupling, so in a way, this would seem as a system that can, orient spontaneously and randomly in different directions. However, what we see in development is that PCP generates very, very consistent and very long-range organized patterns. And there, one of the questions that, to my mind, is still open is really what is the origin of polarity, so what basically nucleates these polarity vectors in specific places, such as to make these patterns reproducible. Secondly, there's the question of long-range vector propagation. And this is, again, think about this as sort of individually polarized units that are coupled by neighbor-neighbor coupling. So actually, one of the challenges in such systems is to avoid the system from getting trapped in local minima, right? And this is, for example, this is one of the important PCP modeling papers by Boris Schreiman, where they demonstrate exactly this effect, that these systems have an intrinsic tendency to sort of get trapped in local minima, and this means that the long-range directionality or these long-range vectors actually start to break down. So clearly this is something that needs to be avoided somehow. And there's a lot of discussion in the field whether this could be happening by gradients, so signaling gradients or protein expression gradients that are superimposed on the field and therefore tend to homogenize the vectors over long distances. Or an idea that was propagated by Frank and Suzanne was that PCP could be established very early on when the tissue is small, and then it's just maintained during growth, so that this is also, again, a possibility of getting around this getting trapped in local minima. And then finally, there's the issue, and this, as a biologist, this to me is the most immediate one. And this is the question of multi-scale alignment of polarity. So again, how do fish, for example, ensure that the scales are oriented along the long axis of the animal and not sort of in like at oblique angles or not oriented at all, right? So in this ultimately is the question of how do you maintain and coordinate these polarity cues from the molecular level ultimately all the way up to the organismal level. And again, this is difficult to study in developmental systems because yes, it's easy to interfere with molecules, but what you cannot do in such a case, for example, is to make a fish that is double-headed or something like this and then ask how polarity, but when you change the polarity at the body plan level, how this influences then the local polarity at the level of cells or within cells. Now, back to the worm, back to planarians. So basically, polarity in our system is also hugely important, and already from this issue that we were discussing yesterday of, if you were to now, yep. Well, that was actually a key theme of Suzanne's work as well, is that when you look at PCP during fly wing development, it is actually very dynamic. And you will see that at different stages of development, the PCP vectors in a specific area will point in different directions. So in this sense, it can be dynamic, it can be reoriented. On the other hand, also in the fly wing, at some point, it becomes frozen because at some point simply the PCP vectors are translated into hair formation. And basically the moment the hair forms, then that's it. So if you were to change the PCP systems there, then it wouldn't have any effect anymore. And on top of that, the PCP systems themselves are highly dynamic because the components are being endocytosed and exocytosed, and they interact with the cytoskeleton, and there are cause-consequence relationships there where you need polarized cytoskeleton to get polarized PCP, but again, you need polarized PCP to get polarized cytoskeleton. And so, in short, lots of dynamics in the system, but what often happens in development is that at certain stages it becomes frozen and is then used as a stable vector from then on. Okay, so back to planarians. Again, issue, we cut a circle out of the center of the animal and we watch it regenerate. Question, does this little circle that's cut out of the animal, does it remember its original orientation in the worm? Or will it establish the new head and the tail sort of randomly, de novo, somewhere along the perimeter? And now one simple way of how you can ask this question is, for example, by cutting a triangle out of the head of the animal. So right in both cases, basically the shape of the tissue piece that regenerates is exactly the same. It also comes from the same body area of the worm, except that in the first case, the flat side of the triangle is facing forward, whereas in the second case, the tip of the triangle is facing forward. Right now, if there were no polarity in the system, or if in other words, the piece would randomly initiate the head somewhere and the tail somewhere else, then there shouldn't be a difference between these two scenarios. However, what we observe is that invariably, with such cases, the new eyes always form on the flat side of a triangle, whereas in the case when the tip is facing forward, the new eyes always form between the points of the triangle. And so this is, just a very simple way of demonstrating that indeed planarian tissues are intrinsically polarized, and that this intrinsic tissue polarization also ultimately instructs the polarization of the regenerative processes. And so this, in fact, has been known since a long time. So T.H. Morgan, the famous Drosophila geneticist and also like Nobel laureate, he worked actually on planarians before he got sidetracked with Drosophila. And in one of his papers, he states that something in the piece itself determines that a head shall develop at the anterior cut surface and the tail at the posterior cut surface. This something is what we call polarity, right? And I will also stick for the remainder of the talk, I will stick largely to this definition of polarity. And then, of course, the second asset that the Planarian system brings to the table of this entire issue of thinking about polarity is that indeed in this system, we have the body plan polarity as an experimental variable. Right, so as I've shown to you yesterday, we can make these double-headed animals and we can make double-tailed animals. And today the movies even work. So for this reason, we can, in a way, you can think of this as we can reprogram global body plan polarity. And then we can ask in this system, what are the local consequences of such changes at the global level? Now, but in order to do that, we, of course, need to have a way of visualizing polarity in the Planarian system. And if you were to work in a mouse or in a fly or in a zebrafish, this wouldn't be a problem at all because you would have 1,000 markers. But in these non-conventional model systems, something like this is already a challenge, right? How do you build a marker for polarity in the system? And what we decided to do was to make use of the motility of planarians. And so what you see in this movie, if I play it again, is essentially worms. So this is about 3 times sped up or so. And you see the worms just gliding around in their culture dish. And they move smoothly by just gliding over the surface. And what allows them to do this is actually that their ventral epidermis, so the belly side, is densely ciliated. And this is, so in a way, this tissue is very similar to the lining of our lungs, for example. So it consists of many cells that all contain many, many cilia. And as I've shown to you before, well, I basically argued before, in order to generate directional motility on basis of cilia, the cilia need to be organized or need to be aligned. So they need to be polarized along a certain axis. However, if you just look at cilia, so these long and flexible axonemes, then it looks like fur, and you cannot infer any polarity from such a staining. However, each cilium itself is anchored within the cell with a highly polarized structure. So basically, each cilium terminates within the cell in a basal body. So that's basically the seat that makes the cilium. And on one side of this basal body, there's a long extension. This is the so-called rootlet. And then there are other parts that stick out in the opposite direction. And actually, this is also the case in planarians. So here we are looking at an electron micrograph of a grazing section of the epidermis. So basically, we are just slicing through the epidermis in the plane of the epidermal cells. And what you see here, these long structures, they are exactly these representations of ciliary rootlets. So here with the basal body, and here with the striated rootlet extending distally. And importantly, already in this electron micrograph, you can see that the rootlets are actually fairly well oriented within an individual cell, and we can also see between neighboring cells. So what this means is that we can use the ciliary rootlet orientation in the Planarian epidermis as basically a readout of polarity in the system. However, there's a catch to that, and EM is great for visualizing things, but the field of view tends to be tiny. And so for our purposes, we of course would like to see rootlet orientation in the entire epidermis. So therefore, what we did was to raise antibodies against the rootlet protein, this long extension, and against the component of the basal body. And so what this does is, sorry, this is difficult to see, is basically that it now labels each rootlet with a green, as basically sort of a green extension with a little red dot at the end. And it's a bit difficult to see here, but even at the level of the light microscope, we can resolve, therefore, a polarity vector within individual rootlets. And then in addition, we developed an imaging pipeline where we use fast spinning disk Z-stack imaging. We then extract computationally the surface of the animal, so basically the epidermis, and then we build a set of tools or a set of algorithms that allow us to first detect individual rootlets and then also assign polarity vectors for each individual rootlet on basis of this offset between the red staining and the green staining. So long story short, what this gives us is in fact a method in which we can visualize polarity at all scales of organization, from the subcellular level on the basis of individual rootlets to individual cells, where we can then generate polarity vectors by averaging the rootlet orientation of all the rootlets in the cells, to the local tissue environment, so for example, by averaging individual cellular polarity vectors now within a given area, and then ultimately all the way up to the entire animal, because we, like with this pipeline, we can image and process like entire animals that have thousands of cell diameters along their AP axis. So this then really gives us a method to visualize the organismal polarity field in the epidermis of the guys. Now, nice and good, what do we see? What we see is this. So this here is essentially one of these stained preparations. What you see protruding out here is the pharynx, so basically again this long muscular feeding tube. And so basically all this here are the epidermal cells, each of which contain about 30 to 50 individual cilia. And so this image is at a much lower resolution, but we basically image this entire thing such that we can resolve individual cilia. And what the representation up here is basically now averaging about 100 by 100 micrometer squares and averaging the orientation of individual cellular polarity vectors within that to generate then a single arrow. And what you can see immediately by looking at this representation up here is that overall, indeed, the cilia in the animal are highly organized, right? So it's obvious that the field is actually quite coherent. On top of that, there's clearly a strong AP orientation component. So this is the head, this is the tail, and cilia are really nicely oriented along this head-tail axis, which absolutely makes sense, right? Because this is the direction in which they move, essentially. But on top of that, what you can also see here, especially in the head of the animal, is that cilia, especially more towards the margins, are deviating towards the side. And so we can quantify this. And so I'm going to show you a lot of these plots where basically here there's 0 means the orientation of cilia straight along the AP axis, and then deviations towards this side. So basically mean increasing angles, and 90 degrees would mean oriented at right angles to the midline, like so, or minus 90 degrees would be oriented at right angles in this direction. And then if you now plot the angles of individual cilia on a trajectory from the left end or from the left side of the animal all the way across to the right side of the animal, you can actually see that in the head there's a significant deviation towards the sides on either end, and that this deviation gets sort of less towards the center of the animal, and it then almost disappears in the tail. So basically this means that the field, this polarity field, is also under the influence of a mediolateral component, and that interestingly, this influence is actually graded along the AP axis, so most strongly felt in the head and less so towards the tail of the animal. Now, this then, raised, so having this field now raised the question of what actually determines now the polarity of the field, or what are the cues that give the orientation to the epidermal polarity field. And I mean, when you think about this naively, you could think, for example, that the head would be orienting this polarity field, but it could also just as well be that the tail would have a key role in orienting the polarity field. And here, of course, now these double-headed or double-tailed animals come in very handy, because now we can ask if, for example, the head were the determinant of the AP orientation of cilia, then in this case, we should get a bipolar polarity field. Whereas if the tail were the determinant of the cilia field, then we should also get a bipolar polarity field, but now pointing in opposite directions. Mm-hmm.
Two questions.
The polarity, to some extent, yes, I will show some of this data later.
Moving.
You mean visualizing the cilia under this
condition? The vectors that you drew.
Nope. We cannot do this in live, but I mean, there are ways of doing it. And actually, there was just a paper on the or put on the archive where they looked at the worms that were sort of gliding on the meniscus of the surface of the water, and then they put tiny beads on the worms. And so they can also, from this, they can then see flow fields. So this could be a possibility to look at the, the local flow.
Or may not be related to the
Exactly, but this is exactly the question we are testing now, right? To ask if we now, so if we change the polarity of the organism, does this then feed back to the local polarity of the cilia and so on and so forth? Because yes, one possibility could also be that actually none of these changes have any effect. But because there is this coupling generally, or the alignment between cilia along the long axis, in some ways there has to be some coupling in order to get this reproducibly aligned along the long axis. Yes, well, they are predators actually, so they will catch, crustaceans and things. But you can also, for example, put a lump of food in the dish and they will all run towards it. Okay, so multiple possibilities, right, of what we could see, and including the null hypothesis that nothing changes when we change head versus tail at the organismal level. However, the answer is that when we look now at the double-headed animals, what we do see are in fact very nice and very clear bipolar polarity fields. So as you can see here, on either end, actually the vectors point inwards, like in a wild-type animal. Moreover, the animal is basically almost exactly divided in half. So each one of these fields now reaches to the center of the animal, and there are, interesting sort of polarity defects, or basically the way where the two fields collide, how the field deals with these opposing polarity vectors. So from this image alone, it's already clear that changing the polarity at the body plan level does feed back on the local orientation of cilia. And on top of that the head clearly is providing a long-range polarity vector to the cell field. Now, what about the tails? So, interestingly, when we look at the double-tailed animals, we see exactly the same thing, except that in this case, the polarity vectors now all point outwards. And interestingly, in this case, also the transition zone in the center looks somewhat different, as in the case of beta-catenin. And so this is also something we are trying to understand at the moment, what's behind that. But so now what this means is that, in fact, both the head and the tail, they both provide long-range polarity vectors to the cell field, But because they basically orient polarity in opposite directions, they act synergistically such that you can think of the head as repelling polarity, whereas the tail attracts the polarity vectors of the epithelium. And now, of course, an interesting question in this sense is to ask what happens if there's neither a head nor a tail. And because planarians are an awesome system in this sense, we can also address this question by knocking down a specific transcription factor that is basically required for making the head and the tail organizing centers. And if you do this, you can see that the molecular markers for the organizing centers are gone. And these animals are essentially headless and tailless, and the ends of the pieces, so to speak, they are basically radially symmetric because there's no more midline in there. So long story short, I mean, the details here don't matter. We can simply use this trick to generate headless and tailless animals. And what happens in these animals is also very interesting because, first of all, the polarity of the system is actually rather well maintained. And this in itself is already interesting because it means that there's at least some sort of, I don't know how to think about it, like, or what's the correct term, but some degree of inertia in the system, that it basically can maintain consistent orientation in the absence of head and tail as organizing cues. But in addition, what is very interesting in these animals is that when you look closely at the ends, the rear end of the piece, so where the tail would have been, this is generally fairly normal. However, at the front end of the piece, where the head would have been normally, what we invariably see there is actually a local reversal of polarity. So basically, at the extreme, at the edges, the cilia point in the opposite direction. They point out rather than in, as they should at a wild-type head. And this then gives rise to this type 1 defect here, where the field then has to compensate somehow. And this is, first of all, interesting because it means that possibly the body edge could act as an attractor for cilia. And secondly, what is interesting is that you only see it at one end, not at the other end. And one way of rationalizing this is to think that exactly what we are dealing with is basically a universal attraction of the body margins, so in black, that they basically tend to orient cilia towards the outside. And under these conditions, in the tail, this effect would actually largely synergize with the direction of the polarity field in the existing piece, whereas in the head, it would then generate a conflict with the existing polarity field. And so according to this way of thinking about it, actually what this data could indicate is that the body edge indeed acts as some sort of universal mediolateral cue to attract cilia orientation. Yep. Yep, exactly. But in this case, we can show molecularly that there's really no tail here anteriorly. So when you look for all the genes that are normally expressed in a tail, you don't find them here. And likewise, when you look for the head genes, they are not expressed here. So this would have to be a generic effect that is not tied to the tissue identity, tail or head. You can get it also with double-headed or double-tailed animals if the new.
In the headless.
No, this is interestingly, this is always at this position. Yes, and but we will get back to this point later, or at least how we think about it. All right. So now, and finally, a really important question is, so now we can change the polarity field, but now what are the dynamics of this polarity reversal? So we basically know that in these double-headed animals, right, so at the late stage, after about 2 weeks, we will have these bipolar symmetric polarity fields, whereas at the very starting point, so when we first amputate the piece, then the polarity will actually be consistent across the entire piece. So therefore, to document how we go from consistent orientation to bipolar polarity field, we simply did a time course experiment, so where we just took these pieces from beta-catenin RNAi animals at different time points, and we simply asked at what points and in which ways do we start to see the polarity reversal here at the rear end that normally should have made a tail, but now under this RNAi condition will make a head. And the data is actually surprisingly clear. So what we see is that, interestingly, for the first 3 days of regeneration, actually nothing much is happening, and the orientation within the pieces is rather much consistent along the long axis. And this is interesting because we know that molecularly, this wound here will already be specified as a head regenerating wound by about 20 to 24 hours. So, and instead, in terms of the cilia orientation, nothing much happens until day 3. So this already means that there's actually a significant time lag in the system. But what then happens is that over the subsequent days, we actually start to see the polarity reversal at the distal end. So first at about 3 days post-amputation, where you see that the polarity now switches to the opposite, to basically 180-degree orientation. And then over the subsequent days, what we see is that basically this reverse orientation now starts to spread into the tissue. So even though this is a rather crude experiment, and we would of course love to have this in much more dynamic detail, what this experiment shows is that the polarity reversal actually initiates at the boundary, so at the tip of the future head, and then from there it propagates in some sort of a wave-like manner into the tissue. Basically the propagation also over time gradually slows down until it comes to a halt in the geometric center of this tissue piece here. So in this sense, so this would be a case where really we have initiated some sort of a symmetry breaking event at this end, and where the consequences of this symmetry breaking event then propagate into the cell field and change the polarity of the cell field in the process. Now, putting all of this together, we decided to formulate a mathematical model together with our collaborators at the TU Dresden this time. And basically this model is essentially based on the biological observations that I've shown you so far. So the first assumption is that cells are individually polarized, that the polarization of individual cells propagates to their neighbors via neighbor-neighbor coupling, and that on top of that, the head and the tail oppositely orient the polarity vector, but thereby together create basically consistent orientation along the AP axis. And that finally, fourth, we are dealing with separate polarity fields. One that orients along the AP axis. So this is what's mediated by the head and by the tail. And the second field is this sort of where we assume this radially directed attractive effect of the margin. Now we think, or that this is the assumption of the model, that the final polarity as we observe it by example of the cilia orientation, that this is actually the result of the superposition of these two polarity fields. So first of all, the AP polarity field mediated by head-tail. Second, this sort of generally globally attractive effects of the boundary. Now, the model is basically inspired by Frank's and Suzanne's boundary model work, but we explicitly coarse grain it. So we don't model any details about the actual polarization mechanisms within the cells, also simply because we don't know them. Instead, what we assume is that the polarity vector is specified at the boundary, and from the boundary propagates into the cell field by neighbor-neighbor coupling. And so we can then simulate, or we can then basically model the change in polarity at a specific place via the set of partial differential equations that basically include the specification of the vector at the boundary, basically via localized reaction conditions at the boundary. They include a noise component, which is due to the dynamic replacement, or we assume to be due to the dynamic replacement of cells, and so on and so forth. A spatial averaging Laplacian, and then the simple linear superposition of the two cell fields together. And with this model now, we can ask, the first thing we did was to simply ask to what extent, with this rather simple model, can we actually recapitulate the experimental observations. This is assumed to be just a noise term here. So, and the noise would be separate, so that basically introduces some sort of uncertainty into the alignment of the coupling process. And we assume this to be separate for the head-tail component and for the mediolateral component, because we assume that they are different mechanisms. Okay, and then basically what is interesting is that this model actually reproduces display effect in wild-type animals, simply because, again, in the tail area of the animal, the effect of this, the radially directed effect of the boundary is in alignment with the anterior-posterior directed effect of the head-tail field. But in the head, the two are actually in conflict. Second, just by simply turning off head and tail, always with the same simulation parameters, we can show that we can reproduce the double-tailed and the double-headed effect. This is not so amazing. But interestingly, we can also recapitulate this local polarity defect in the headless, tailless animals. However, in the model, it is recapitulated as a temporal intermediate, because if you let the simulations run, this defect would actually propagate towards the center of the animal, whereas this is not what we observe in biology. So here we can qualitatively recapitulate the results, but quantitatively model and reality diverge somewhat at this point. But to me, actually, the most interesting result was that the model also recapitulates the dynamic spread of the polarity wave from the boundary towards the center in these time course experiments that I've shown you a while ago. And so basically, if we measure here the position of the wave in the tissue as a function of time, then, and this is, so the measurements are shown in these colored plots down here, then what you can see is exactly this effect that I mentioned to you before, that basically the speed of propagation of this wave slows down over time until it comes to halt in the center of the cell field. And now, interestingly, if we simulate this reversal by just basically turning a tail pole into a head pole and then asking how this effect propagates into the tissue, we see a remarkably similar propagation kinetic. So this is the black line here, which overall matches the measured points relatively well. And this is due to the diffusion term that we assume to mediate the neighbor-neighbor coupling of the boundary condition into the field, or the propagation of the boundary condition into the field, because this then will tend to become weaker and weaker the further away you get from the boundary, which then also results in a slowdown of this propagation here. So long story short, this is indeed a rather simple description of this polarity field of the Planarian epidermis, but nevertheless, it at least represents quantitatively some aspects of the polarity field that we do observe in experiments. Now, the question that was interesting me a lot, and also my people, was of course the question now, what are the molecular mechanisms that mediate the polarization of the Planarian cell field? And secondly, if we were to know the molecular mechanisms, could we then in some ways validate this model, or could we refine the model on basis of these molecular mechanisms? And specifically, our model, or the way we model polarity, this would make two specific predictions. And the first one is that we should have distinct molecular mediators of this ML and AP polarity field, right? Because it is the superposition between the two fields in our model that generates the final cilia alignment. And second, then as a consequence of that, we would predict to have uniform AP polarization when this mediolateral component is missing. Or secondly, we should have loss of AP polarization and basically radial orientation of the cilia if the AP component were missing. And now, in order to find the molecules that do this, we use sort of the standard molecular biology approach. And that is that we first went into the genome, into the Planarian genome that we sequenced, and we simply asked, what components of the piece of known PCP machineries can we find in planarians? And there actually the answer is that pretty much all the PCP components that are known from Drosophila or also from vertebrates are also found in planarians. Now this in itself is not very surprising because again these standard molecular mechanisms, they get reused over and over again in evolution, so you basically find them in all animals. And secondly, what we then did was to do an RNAi screen, so for these components. So we basically remove the gene function of PCP components in planarians and then ask for effects on, or well, basically screen for effects on the global polarity field of the animals. So again, so if the green component, the radial component were missing, we would predict that such genes should give us uniform AP orientation, whereas in the other case, we would expect to see radial orientation of the cilia. Now, in this sense, we were very delighted when we knocked down this component, so DAXs. So this was the second PCP pathway that I introduced you earlier. This is a component of this fat DAXs PCP pathway. What we find is pretty much exactly this. So now all along basically the cilia are highly consistently oriented along the AP axis, and we don't have display anymore as we have it in wild-type animals. This we can also show quantitatively. So essentially here in black, this would be the curve from wild-type animals, and it basically deviates from zero, so straight AP orientation in the head, because that's where the cilia are pointing outwards. And then over the long axis of the animal, display effect gets progressively less. Whereas if we now quantify DAXs RNAi animals, what we find is that display is pretty much uniform all along the AP axis. And this we can also show that this is quantitatively the case and statistically significant different. So what is clear therefore from these data is that fat DAXs is clearly involved in the orientation of cilia towards the body margins. Sorry, question? They are actually, when you watch them, it doesn't seem to have any consequences on their movement. So they go nice and straight. Now, but to test to what extent, again, this relates to our model, we decided to turn to these headless animals again. And remember, so in the headless animals, we surmise that this defect at the anterior end specifically is due to the fact that here, the attractive effect or an unattractive effect from the margins is generating a local conflict with the predominant tissue or polarity orientation in the tissue. So if this were the case, we would predict that in such a scenario, if we were to remove the radially orienting Q, that then we should actually rescue this anterior polarity defect. And so this is exactly what we did then by basically combining the knockdown of DAXs, so this component that's required for the radial orientation of cilia, with PBX, so the thing that makes the headless, tailless animals. And if we do that, then we find that indeed the double knockdown rescues this type 1 polarity defect. And so this here again in dashed now is showing the situation in wild type animals, where you can see that essentially what this curve shows is that up till this point in the tissues, the cilia are actually pointing in the opposite direction, so are pointing outwards. So then until they switch to the predominant orientation in the rest of the animal. Whereas in the double RNAi animals, what we see is that either this zone of polarity reversal close to the margin is either very much shortened or actually not visible at all. So almost all the way out to the tip, the cilia are pointing in the same direction. So and on top of that, this molecule Daxus is actually nicely expressed where we would want it to be expressed, namely all around the body margins of the animal. So overall, therefore, this is pretty good evidence that fat Daxus indeed mediates the ML polarization component in planarians or in the Planarian system. Now, what about the AP orientation component? And again, in this case, we would expect that genes that affect the AP orientation, if we remove them from the system, we should end up with more or less radially oriented cilia. And indeed, we did find such genes. And so in this case, for example, disheveled 1,2, or basically we have to knock down the 2 disheveled homologs, that's why disheveled 1,2. This gives very beautifully, well, or very convincingly actually, this radial reorientation defect as predicted. And moreover, we see exactly the same thing if we knock down these Wangel components. So, and yeah, sorry, that isn't mentioned here. So both of them are, of course, part of the canonical PCP pathway. So moreover, again, we can show that whereas wild-type animals tend to have this predominant AP orientation, essentially these disheveled animals invariably have cilia that almost point towards the outside of the animal consistently all across the length of the animal. And again, this is also highly reproducible and statistically significant. So therefore, from this, it's clear that core PCP or the core PCP pathway is actually required for orienting cilia along the AP axis of the animals. And in this sense, then, a fun thing to ask was, okay, fine. Now, if disheveled or canonical PCP mediates orientation along the long axis, and this fat Daxusing mediates the radial component, what would happen then if we take both away? Any guesses? Come on. Random? One? No effect. Null hypothesis, always good. That's exactly what I was hoping for. But instead, what we got was, in fact, something very close to the no effect, where now we know that both pathways are inactivated for other biochemical assays that I'm not showing you. However, nevertheless, the orientation of cilia in the animals is not entirely wild type because there's a lot more variation between animals, but it pretty closely follows the mean orientation in wild type animals here. So, and in a way, this is nice because actually we would, so also from our model where the cilia orientation is basically the superposition of the two independent polarities, with the model we can rationalize this rescue as basically saying that if you weaken both components, that then again you end up with something that is actually pretty close to normal. However, in terms of thinking about polarity and what causes polarity ultimately, this is deeply puzzling because if by taking away both polarity machineries, you end up with actually a nicely polarized cell field, then this raises the rather profound question of what polarizes then the cell field, if not the polarity machines. We will get back to this in a little while. Yep?
can understand that if you do pathway, go to both, still get the same.
So we have to go into the model, and also I have to read again the details of it. But we have two parameters that mediate the orientation. So first, the boundary effects in the end, but then also the sensitivity of the responsiveness of the cells to these orienting effects.
So the orienting effect at the boundary for every.
No, they are only at the boundary. So basically, we specify the polarity vectors at the boundary, and from there they get propagated into the cell field. But there's a second parameter in there that basically determines the sensitivity of the cells to the two fields. But we need to look at it again because we submitted the paper a year ago. Okay, and yeah, so this is essentially then, the model of how we, or at least the biological model of how we envisage polarity in the Planarian system, so that we think that essentially every single cell, as in the Drosophila system or as in other systems, is intrinsically polarized. But importantly, it is intrinsically polarized along two directions. So first, along the head tail, along basically along one axis, so here red and blue, and this is mediated by PCP. Second, it is polarized along the mediolateral axis, and this effect is mediated by fat axis. And third, the orientation of cilia within the cell, so here shown in gray, they basically orient along the resultant of these two different vectors within each cell. Now, at the whole organism level, these two vectors are oriented by the tip of the head and by the tip of the tail, in the case of the canonical PCP system, and by fat axis in case of this margin-oriented PCP field. And then sort of at the tissue level, cells, or basically these margin effects, they get propagated via this neighbor-neighbor coupling to neighboring cells, as is known from these mechanisms in Drosophila. Now, in a way, this is nice because we can say that at least we can rationalize with this model how polarity might be, or how this coordination of polarity might be achieved between the organismal level and the cellular level. On the other hand, as always, probably with biological models, especially in planarians, this is of course very, very coarse-grained and leaves many important mechanistic questions. And so the first one and very immediate one is that we need to, of course, visualize the polarity complexes within the cells in order to show that the cellular component of this is actually happening. And on top of that, there are a bunch of other interesting questions or other important questions that I want to go through now. Yeah, sorry. And what I forgot to mention is that this scenario or this topology and also the likely mechanisms of PCP orientation in planarians is very reminiscent of the Drosophila wing disc, where, as I already said in the beginning, there's also a predominant influence of fat axis that comes from the margins and basically orients the developing wing cells in a radial direction. And then there's canonical PCP, which tends to orient cells along the long axis of the future wing, so towards this boundary here. And so again, this is basically reflecting very likely simply the reutilization of these pathways in evolution, and therefore shows that via these two pathways, you can generate actually 2D effects in polarity fields in biological systems. Now, as to the open questions, so the first, from a biological point of view and also from a conceptual point of view, key point is what are the poles, what are the ends of the animals actually doing to this PCP vector? So that is basically the question of what is the origin of the symmetry breaking event, or what orients the cell field from the boundary. And here actually one interesting idea or one interesting bit of data from Drosophila is that in Drosophila it has been hypothesized, and some people claim that it has been shown that actually gradients of wind proteins are responsible for generating the polarity vector for canonical PCP in the Drosophila wing disk. And this is based, again, remember that the wind signaling pathway and this canonical PCP signaling pathway share components. So amongst them, the receptor for the wind ligand per se. And so one group has shown that by providing a source of the wind ligand, that this alone is sufficient for generating long-range orientation of the PCP components within the cell. Now, in our system, this model is interesting because, as you know from yesterday's talk, we have in fact many wind ligands that are expressed along the long axis of the animal, so that are high in the tail and low in the head. So this could indeed provide, some sort of molecular or be part of the molecular explanation as towards the orienting effects of the tail. However, then from our data, we would also have to surmise that there's a similar effect that is mediated by the head, because both head and tail can mediate polarity independently, as we've shown. And this, there's no example in the literature of a mechanism that could account for this, because that would basically have to be something that targets Wangel. So that's a very interesting point we are going after now molecularly. Secondly, is this question of, why do we not see swirls in this system? And this is one that I find deeply interesting, again, because of this effect or of this danger of getting trapped in local minima. And now just remember again that planarians are basically at least an order of magnitude larger than a Drosophila wing disk. And so if, there would have been some sort of this length dependent, or like if this were a general problem, that the larger the cell field, the more likely that you get some sort of local trapping effects or local inhomogeneities, spatial inhomogeneities, then if there were a system where we should see it, then for sure in planarians, given that they are so much larger. But instead, we don't. Yeah. So I think ultimately it would depend on the, on the number of cells, right? Because each cell basically acts as unit of polarity that can polarize itself and can then couple to its neighbors. So I would have said, because the distances in planarians are very large, so meaning we have many more cells in between the orienting, the regions that mediate the orientation. Therefore, it would have been more likely to see a breakdown of this long-range orientation at some point in the cell field. But instead, we don't. Sorry, is this clear? Sorry, wing hair, you mean? Sorry, I don't understand. I think we... Latter, the latter. We need to discuss this later. Yeah.
Vectors, single cell.
Which vectors?
The vectors that you're drawing there.
Yeah.
Is from looking at single cell orientation. The planning is to 100. World size is actually smaller than that.
We also looked with higher resolution at smaller cell fields, and we don't see an effect there either. So one possibility could be again related to this idea up here that gradients are actually important for orienting polarity, right? So we know that in planarians there are these long-range signaling gradients, so it is possible that they present an orienting influence. Another possibility is that actually the epidermis of planarians, as I've shown you yesterday, is highly, highly dynamic. So the cells, the polarized cells themselves, turn over continuously via new cells integrating from below. And so presumably what this requires is that a new cell that integrates in some sort of a naive state and then polarizes according to the average effects of its neighbors. So this could be also, as our collaborators have shown in this paper, so such dynamic turnover of the constituents of cells in planarly polarized tissues can actually also make the probability of getting trapped in these local minima less. And finally, there's this question, or that I find still deeply interesting, and this is, if it is not PCP and or fat daxis that actually orients the cilia and the wing hairs, so what is it then? Or why, in other words, if you take both systems away, why do you still get oriented cilia or oriented wing hairs? And by the way, Suzanne has done this experiment in Drosophila much more cleanly because they have genetically removed both systems. And also under these conditions, there are still beautiful, consistent wing hair orientation, not like in a wild type wing, because also the wing has sort of different shapes and things like that. But nevertheless, there is still consistent orientation of wing hairs between neighboring cells. And what this basically shows is that the PCP machineries, they cannot be responsible for polarity. All they are doing is actually guiding some other polarity process into predictable and genetically determined orientations. And this then, of course, raises the question of what is this other polarity process? And to my mind, this is a completely open question. So what we are thinking about now is that actually maybe the ciliary rootlets or the wing hairs in Drosophila, maybe rather than passively aligning to the PCP field that is generated by the polarity machineries, maybe they are actually generating the polarity themselves. And so, for example, in the case of the ciliary rootlets, because they are all individually highly polarized structures, you could, and we know that they interact with the cytoskeleton in predetermined ways, you could actually think, and this has been shown in some systems, that the ciliary rootlets of a given cell polymerize into some sort of a grid via regular head-to-tail arrangements. So you could imagine that via this rootlet polymerization into grids, you actually generate polarity at this level that then can get oriented via the junctional PCP complexes. And also in the Drosophila wing, so something I've been discussing with Suzanne a lot was the possibility that maybe actually the act of wing hair outgrowth itself has a polarizing effect on the neighboring cell. And this is very well imaginable because the wing hair is initially a solid rod of actin filaments that pushes out almost in the plane of the epithelium. So it will almost certainly have a mechanical effect on the neighboring cell. And I think also that now, and because how much time do I have left? Oh, very good. So then so now finally, back to this question. So we've been talking a lot about polarity now. So is everything that I've been telling you, is any of what I've been telling you today now, is this relevant to this question? So to the intrinsic tissue polarity in planarians. And here, to our great disappointment, we have to actually say no. Because when we take away the components of the canonical PCP system that, as you remember, we know are responsible for orienting cilia along the AP axis, then unfortunately nothing happens to regeneration polarity at all. So these animals make beautiful heads, they make beautiful tails, and everything is just fine. So what this means is that in the grand scheme of things, with respect to explaining Planarian regeneration polarity, we were unfortunately following a red herring. But nevertheless, in this case, at least, it also led to interesting insights into another phenomenon that was not primarily on our original target list. And for the rest of the talk, I actually wanted to tell you about our current attempts or ongoing attempts to test whether or not this wind signaling gradient itself could be the polarity cue. But also after 2.5 years, we similarly came to the conclusion that this is also a red herring. So for this reason, I think we can easily skip the second part and simply finish here.
Culture planaria in an environment with specific agents. Is it possible that I change the polarity of cells?
You mean specifically of individual cells without affecting the polarity of the whole animal? Or?
Yeah.
Well, I mean, we haven't tried it, but I would guess if you, depolymerize actin filaments or microtubules, that you could affect cilia the arrangement of cilia. Possibly. And then again, the question would be, does it recover? Probably. We haven't done these experiments, but that would be very interesting.
The other question. In one slide, you showed a neighbor polarity, which polarity field was head. Tail neighbor or just head?
Sorry,
there was a picture that showed the polarity field was based on if the polarity field was such a situation that in both sides we have head. In both sides we have sources of I mean, there is situation that we have neighbor, but in both sides we have tail, or not.
So to have double-tailed animals.
Neighbor, double tail or double head.
In neighboring cells, or like in cells that directly touch each other.
Yeah.
We would predict, no under ordinary circumstances because of these various feedback loops that are involved. But to really test that, we would be able to see the protein components directly, and this we can't at the moment.
So actually, I have a couple of comments and a couple of questions. So the first comment is that coming to very similar points he raised, there are multiciliated systems. There, the spacing of cilia is dependent upon some other process. Can that be also accounted for the things which you are looking at? Because there also there are rootlets and there are distal and subdistal fibers which are very long, and they actually help in determining proper spacing between each cilia. and also the polarity in them. The other idea was that if we are thinking it on the basis of cell autonomous driven polarity generation, then it could be that the Wangel frizzled pathway or is just tightening up the polarity in AP axis, and the fat dusters pathway is tightening up the polarity in ML axis. So when we are removing proteins, we are removing just fat, not just dusters, or you are removing just dusters or fat. But if we remove both of the component of the same pathway, both fat and dusters from the same embryo, what do you expect? That if you remove this, you will get the AP axis in a very symmetrical fashion, but in the ML axis, I will expect something which is very much similar to what you have seen in the when you have removed Wangel and fat or dashes. So my idea is that if you remove both the component of one polarity determining axis, like in ML axis, we remove both fat and dashes, you will see the AP axis is very rigid polarity wise, but on the ML axis, you will see there is deviation. Contrary to that, if you remove Wangel and frizzled both from the system, you will see that the ML axis is fine, but the AP axis, there is problem in polarity.
I'm just trying to think which of these experiments we've done actually. But let me start with your first question. So basically the question of to what extent the known intracellular alignment of cilia or rootlets, to what extent this could be part of these mechanisms. And first of all, this is essentially what I'm thinking about when I'm thinking about this polymerization of rootlets into a grid, right? So that would be exactly the interaction also between neighboring basal bodies and in a head to tail and in a lateral manner via all the cytoskeletal pathways that have been shown to play a role in this case. However, this can explain how to orient cilia within a single cell, right? But it doesn't per se explain how you can then couple the orientation vectors between neighboring cells. And of course, one really interesting possibility there is what has also been shown, right? That the flow that is generated by cilia, that this in itself feeds back on the alignment of cilia. So in this sense, it could be that just by orienting cilia in individual cells, you could then have this neighbor-neighbor coupling effect as a consequence of the flow that this alignment generates. However, these experiments are also hugely controversial because even in Xenopus, where it was originally shown, they are very difficult to reproduce. Now, the second question of whether we see the same or different things when we take out just one or both components of a pathway. So I'm fairly sure that we've done it for fat and DAXs, that we knocked them both down, or actually three because there are multiple fats. And the effects are essentially indistinguishable from just knocking down DAXs alone. So for the AP part, for the frizzled and Wangel, it's much more difficult because there are about 8 different frizzles in the system. So we first of all, we don't know which one is responsible for PCP. And second, it's very likely multiple. So we would have to take out like all 8 of them to see something. And then in our current system, all sorts of other things go wrong. So we simply cannot do this experiment at the moment. But it's an interesting idea. So because also head and tail in a way are both mediating long-range effects in opposite directions, but by coupling to the same pathway. So for this reason, I would also predict that there should be differences in the molecular phenotypes.
Animals? Other questions? Right, so let's take a break. Let's go for lunch and come back at 2 for Matthew's talk.